What's up guys, good evening my chilla party. This is your NSEJS Biology Master Teacher Akshansh and welcome back to the session number 4 for NSEJS 2025 Biology Basic to Advanced series. I hope you would have watched the last 3 lectures where we had started exploring about biomolecules. Well, if you have, great, we are all set for today's discussion, but if you have not, I'd highly recommend catching up to make most out of this session. Now, we will continue with the RNA. So let's move on to the particular page. So now let's move on and let's continue with what is RNA. So RNA stands for ribonucleic acid. What is RNA? Well, RNA stands for ribonucleic acid. Okay, so RNA stands for ribonucleic acid. So as the name suggests, ribonucleic acid, it is formed by end-to-end polymerization of a number of ribonucleotides or simply ribotypes. Okay, so it is formed by end-to-end polymerization of ribonucleotides or simply ribotypes. Yes. So ribonucleic acid formed by end-to-end polymerization of ribonucleotides or ribotypes. Now I will tell you how the RNA is formed. According to central dogma, RNA is transcribed from the DNA and is further processed into various types of RNAs. Okay, so RNA, we have the template DNA. The template DNA gets transcribed. So the transcription happens. Further followed by processing. Okay, we get the different types of RNA. Different types of RNAs. Correct? So let's talk about the ribonucleotides first and then I'll tell you about this particular process. Okay, so first of all, when I say ribonucleotides, what will be they made up of? So ribonucleotides first of all, they are going, since they are nucleotides only, so they will have the sugar. So they will have the sugar that is going to be the ribose sugar in the beta form. Correct? The second is going to be the nitrogen bases. So nitrogen bases mein you have adenine, you have guanine, you have cytosine, and instead of thymine we have uracil. Okay, so uracil, and then we will have the phosphate group. So sugars, nitrogen base, phosphate group combined together will make up your ribonucleotide. So you will have the number of ribonucleotides ranging between 70 to 12,000 in a molecule of RNA. So I will write RNA contains 70 to 12,000 ribonucleotides or simply ribotypes correct? I hope this is clear to all of you. Now let's talk about the classification, like the types of RNA that we have. RNA, okay, so RNA, first of all you have mRNA, that is the messenger RNA. We have rRNA, that is the ribosomal RNA. Then we have tRNA, that is the transfer RNA. Then we have genomic RNA, genomic RNA. Then we have snRNA, small nuclear RNA, snRNA, that is the small nuclear RNA. We have small cytoplasmic RNA, small cytoplasmic RNA, and then we have the regulatory RNA, regulatory RNA, that will further made up of siRNA and miRNA. So here are the types of RNA that you have. So right now I only want you to remember the names of the types of RNA. When we will do the molecular basis of inheritance, or simply the molecular biology, in that particular unit I will teach you about what are these different types of RNAs. Okay, right now you just have to remember the names. So a quick recap. First of all, what is RNA? So RNA stands for ribonucleic acid. It is formed by end-to-end polymerization of ribonucleotides, or simply ribotides. So how the RNA is formed? So we have the template DNA, it undergoes transcription followed by some processing, and that gives rise to different types of RNA. So the composition of the ribonucleotide, so we have ribonucleotide, the sugar, ribose, beta sugar, nitrogen bases A, G, C, U. Instead of thymine, we have uracil in case of in the case of RNA. Then we have the phosphate group. RNA contains 70 to 12,000 ribonucleotides. Then the types of the RNA, so we have mRNA, rRNA, tRNA. mRNA stands for messenger RNA, rRNA that is ribosomal RNA, tRNA that is a transfer RNA, genomic RNA, SN small nuclear RNA, small cytoplasmic RNA, and then we have the regulatory RNA, miRNA and siRNA. So these are the types of RNA that we have. So we are done with the nucleic acids wala part. Now we'll continue with the lipids. So what are lipids? So lipids, basically they are the fatty acid esters of alcohol and related products. What are lipids? So I'll change the color of my pen. They are fatty acid esters. What are they? They are esters. They are not polymers. They are esters. Chemically, they are esters. So they are esters of alcohol, fatty acids ester of alcohol, and related substances. Related substances. Now lipids, so they are insoluble in water. They are insoluble in water, but they are readily dissolved in non-polar solvents. Okay, so I'll write they are insoluble in water but get dissolved in non-polar organic solvents, non-polar organic solvents like, so can you tell me some names of non-polar organic solvents? The first is very common, that is benzene, acetone, chloroform, okay. So what are lipids? Lipids are fatty acid esters of alcohol and related substances. They are insoluble in water but get dissolved in non-polar organic solvents like benzene, acetone and chloroform. Okay, then let's talk about the classification of lipids. What we will talk about? We'll talk about the classification of lipids. Lipids are primarily categorized into three major categories. Okay, so first you can have simple lipids, you can have compound or conjugated lipids, or you can have simply the derived lipids. You can have derived lipids. Okay, so we can have derived lipids. Now let's talk about each and every single type of lipid. So simply simple lipids, they are just simple fatty acid esters. Okay, so they are simple fatty acid esters. So simply the NCERT writes that a fatty acid is a simple lipid. Trihydric alcohol such as glycerol is a simple lipid, and fatty acid plus glycerol is also a simple lipid. So fatty acid glycerol plus glycerol is also lipid. So the neutral fats, wax, the suberin, cutin, these are simple lipids only. So the very popular example are the neutral fats. So the neutral or true fats. What are they? So basically they are triglycerides. Triglycerides. Correct? So next is of the compound or conjugated lipids. So compound or conjugated lipids. So lipid plus you will have a non-lipid part. Okay. So lipid plus a non-lipid part. Correct? And derived. So they are derived from other lipids, or they are chemically like lipids such as sterols. So one example of conjugated lipid that you have is lecithin, that is given in NCERT, and which is important as well. So lecithin, and for derived you have the example of cholesterol. Correct? Okay, so back to the structures first. Let's draw the structure for a triglyceride. So triglycerides. So triglycerides, you will have three fatty acids esterified to a trihydric alcohol such as glycerol. Okay, so first of all, I will draw the structure of glycerol: CH2OH, CHOH, and then again CH2OH. Correct? So this is the structure for glycerol. This is glycerol. You have three OH groups, so trihydric alcohol, OH is alcohol only, and if I have to write the IUPAC name, it will be propane 1,2,3 triol, or it is simply trihydroxypropane. Okay, so now you will have the fatty acids. What are fatty acids? So fatty acids, they are long chain hydrocarbon with the carboxylic acid group. Okay, so here I have the carbon, the fatty acid R2. So what are these? These are the fatty acids. These are fatty acids. Correct? These are fatty acids. So now fatty acids. So we can simply establish the ester bond. So it will look something like this. Okay, so 3 molecules of water will get removed, and you will end up getting a triglyceride which will look something like this. Okay, so this is a triglyceride. This is a triglyceride. So you have fatty acids. Now थोड़ा सा fatty acids के बारे में we can also learn about. Okay, so what are fatty acids? We can write it on the next page. So I said fatty acids. Fatty acids. Fatty acids, long chain hydrocarbon with a carboxylic acid group. So when I say long chain hydrocarbon, so what is going to be the number of carbon atoms that you can have? So book to book the number will vary. I am picking it out from a research paper. The number of carbon atoms can be ranging from 4 to 26. Correct? So in some books it will be given like 4 to 24 or 4 to 28. In some books you will find data like 2 to 22. So the data will vary from book to book. I have picked it out from a particular research paper. So it mentions about 4 to 26 number of carbon atoms that you can have in a fatty acid. Now fatty acid, you can have saturated fatty acids or you can have unsaturated fatty acids. Okay, so you can have the saturated fatty acids or you can have unsaturated fatty acids. Okay, so saturated and unsaturated fatty acids, when I say unsaturated fatty acids, so you are going to have bonds, multiple bonds, double bond or triple bonds, right? Now let's talk about the general formula. So first of all, for saturated fatty acids, it is going to be CnH2nO2. For unsaturated fatty acids, the formula is going to be CnH2n minus 2xO2, where n is the number of atom and x is the number of double bonds. X is the number of double bonds. Correct? X is going to be the number of double bonds. Now we can talk about the saturated fatty acids first. The example, so the very popular example is the palmitic acid and the stearic acid. So the example includes palmitic acid and stearic acid. So for palmitic acid, the value of N is going to be 16 and for stearic acid, N is going to be 18. Okay, so I'll just put the formula. So it will become C16. Here N is going to be 16 and for stearic acid, N is going to be 18. Okay, so for palmitic acid, the formula 16, so it will become C16 H, 16 to the 32, 32 O2, and for the stearic acid 18, so it will become C18 H36 O2, right? Simple. Now let's talk about the unsaturated fatty acids first. So for unsaturated fatty acid, you have to remember the name of 4 unsaturated fatty acids with all double bonds. First is oleic acid, linoleic acid, linoleic acid, and arachidonic acid. Okay, so oleic acid, linoleic acid, linolenic acid, and then we have arachidonic acid. Arachidonic acid. Okay, so now I will also mention the number of carbon atoms they will have and the unsaturation, like the number of double bonds they will have. Okay, so it will be written in the pattern Nx, the value of N and X. So it is for these three, it is 18 carbons, and for arachidonic acid you have 20 carbons, and the number of double bonds 1, 2, 3, 4. Yes. Okay, so I'll tell you, oleic acid will have 18 number of carbons and it will have 1 double bond. Okay, so the formula I told you, CnH2n minus 2xO2. So 18 for the oleic acid, if I have to write the formula, it will look something like this. Okay, so C18H. Now the formula says 2n. So 2 into 18, that is 36. So 36 minus, now how many double bonds we have? So x ka value is 1. So coming back to the formula, minus 2x to minus 2 O2. So the formula comes out to be C18H34O2. Correct? H34O2. Okay. Now simply we can solve it for linoleic acid, linolenic acid, and arachidonic acid. Okay, so it will become C18H. Now 36 minus 2 double bonds, so minus 4, that comes out to be 32 O2 for linoleic acid, right? And for linolenic acid, so 2x, that is 2 into 3, 6. So 36 minus 6, that is 30, right? So C18H30 O2. For arachidonic acid, the number of double bonds is 4. So it is, and the number of carbon is 20. So it is C20, H40 minus 8, that is 32 O2. Okay, so here you have the formula for all these. Now I'll repeat what I have taught you till now so that we can continue. So first of all, we talked about the definition of lipids. What are lipids? So lipids are fatty acid esters of alcohol and related substances. They are insoluble in water, but they get dissolved in non-polar organic solvents like benzene, acetone, chloroform, etc. Then we started talking about the classification of proteins. The classification of lipids. Lipids can be categorized into three major categories. Simple lipids, conjugate or compound lipids, or derived lipids. Simple lipids, so they are fatty acid or glycerol, or fatty acid plus glycerol, the simple lipid. Example includes neutral or trofracs, which are triglycerides. The compound or conjugated mix, so you will have the lipid plus non-lipid part, lecithin example, which is given in NCERT, and then derived lipids. So the derived lipids, they are lipid-like, either they are lipid-like in this structure, example is sterols, such as cholesterol, or they are derived from the lipids only. Okay, then I taught you about the structure for a triglyceride. Okay, so triglyceride means you have the glycerol, and all the three hydroxyl group will be esterified to three fatty acids. Now the fatty acids can be same or they can be different as well. Okay, so this is how the structure, the bonds are established, and this is how the structure of a triglyceride will look like. Okay, this is a triglyceride. Now next, I taught you about the classification of fatty acids. So fatty acids, what are fatty acids? So long hydrocarbons with a carboxylic acid group are fatty acids, right? So you can have saturated fatty acids or unsaturated fatty acids. For saturated fatty acids, you have the formula CnH2nO2, the general formula, and for unsaturated fatty acids, it is CnH2n minus 2x O2, where X is the number of double bonds and N is the number of carbon atoms. So for saturated, you have palmitic acid, stearic acid, and for unsaturated, you have oleic acid, linoleic acid, linoleic acid, and arachidonic acid. Okay, so here are the examples. Now let's move on to the next category, that is of conjugated or compound lipids. Okay, so conjugated lipids. So I will only draw this structure because it is not that much relevant to you. The example is lecithin. The example is lecithin. It is a phospholipid. It is a phospholipid. So as the name suggests, conjugated, it is going to have the lipid plus some non-lipid portion, because it is also going to have a non-lipid part. So lecithin is an example of a phospholipid. So they are found in the plasma membrane. So where you will have lecithin in the plasma membrane or the membranes in the cell. Lecithin ka how the structure will look like. So for lecithin, you will have, again, I'll recall the previous structure for a triglyceride first. So CH2O, C double bond O, R1, CHO, C double bond O, R2. Now with the third group, okay, here you have a diglyceride. What I have drawn is a diglyceride. So here you will have a phosphate group, and to the phosphate group you will have, for lecithin, you will have some other functional group attached to the phosphate group, that is choline. Okay, so lecithin में CH2O, P double bond O, O negative, CH2, CN plus CH3, CH3, CH3. So what you have here is, this is choline. This is choline. Okay. So instead of choline, you can have ethanol amine, you can have serine, or you can have inositol as well. So the name will differ, but this is how the lecithin will look like. Lecithin is a phospholipid. So lecithin is a triglyceride. It is a triglyceride with phosphoric acid, attached to which is the choline group, right? So this is the structure for lecithin. I hope this is clear till here. Okay, now we can talk about the derived lipid, that is cholesterol. So I'll draw the structure for cholesterol over here. Let's draw a line first. Yes. So here we are talking about derived lipids. Correct. The example is cholesterol. Correct? Okay. So cholesterol. First of all, what is the formula? So it is C27H45OH. This is the formula for cholesterol. What it is? C27H45OH. Cholesterol, it is synthesized in the human body. It is synthesized in liver, or also we take it through our diet. Okay. So synthesized by liver, and also it is obtained through diet. Now the next very important point is, so I hope you would have heard about atherosclerosis. When the level of the cholesterol in the blood rises, the cholesterol starts to get deposit in the on the wall of the arteries, decreasing the size of the lumen. Okay, so atherosclerosis, but so that's how it causes problem in human body. But also cholesterol, it is a precursor for the steroid hormones. Okay, so it is precursor for steroid. What is happening? Steroid hormones. So when I say steroid hormones, it is like the testosterone, estrogen, progesterone, etc., etc. Right? We can talk about the structure for cholesterol because it is given in NCRT. Okay, so the structure is very easy. Since cholesterol is a sterol, so it is going to have a cyclopentano hydrophenanthrin ring system. Well, this sounds very complicated, but I'll tell you what it is. So now let's draw the structure for cholesterol because it is given in NCRT. How the structure will look like? First of all, you have uh, two hexagon rings. Oh, this here. So now here you have the structure for cholesterol. This is the structure for cholesterol, right? So this is how you can remember this. Sound looks little difficult, but it is easy to remember. You just have to look at the rings, how the rings are arranged like. So this is the structure for cholesterol. This is what you have to remember. Now we are done with lipids as well. Now we'll continue with the enzymes. What are enzymes? Now enzymes, it is one of the most important topics for NSEJS that you have to prepare by heart. So let's start with the definition first. So what are enzymes? Enzymes are generally proteinaceous substances that are capable of catalyzing a biochemical reaction without undergoing any change. Okay, so enzymes, as I said, what are they? They are proteinaceous substances. So when I say proteinaceous substances, generally they are proteinaceous substances. There are exceptions. Exception. First, may you have ribozymes such as peptidyl transferase. Okay, so ribozymes, they are going to be RNA molecule. It is going to be an RNA molecule. Okay, so proteinaceous substances, exception. The ribozymes, capable of catalyzing a biochemical reaction without undergoing any change. correct? So without undergoing any change, they are capable of catalyzing biochemical reaction. So simply, they are biocatalysts. What are they? They are biocatalysts. They are highly specific. I can tell you the importance of an enzyme with an example. Let's say we have, like, I hope every one of you drink cola or Coca-Cola or Pepsi. So basically what is it? Is carbonated water with some sweeteners, right? So if I have to write a reaction for carbonated water, it will look something like this. H2CO3, that is a carbonic acid, which further uh, ionizes into H plus and bicarbonate ions. Okay, so this reaction also occurs in our blood. Okay, so because of this particular reaction, the blood has its buffering properties. Okay, so now on its own, this reaction, the rate of formation of the carbonic acid, it is 200 molecules per hour, right? If if this reaction is happening without the enzyme, but when you put the enzyme in the presence of the enzyme anhydrase carbonic anhydrase, which is present in the RBC. Okay, so from this 200 molecules per hour, it will become 6 lakh molecules per second. So you can look at the difference an enzyme can create. So how is it achieved? Basically, the enzymes, they provide an alternate pathway, reducing the activation energy for the formation of the intermediates product. Okay, so that's why they are able to catalyze the reaction. Now we can talk about the classification of enzymes. We can talk about the classification of enzymes. So enzymes, you can have a simple enzyme, or you can have a conjugate enzyme. Which is also known as whole enzyme. So simple enzyme, it is going to be completely proteinaceous in nature. It is going to be completely proteinaceous in nature. Example include urease, pepsin, trypsin. Okay, but the conjugate enzyme or an holo enzyme, it is going to have a proteinaceous part and a non-proteinaceous part. Okay, so holo enzyme, it is going to have a proteinaceous part or a non-proteinaceous part. Non-proteinaceous part. Okay, so proteinaceous part, when I say, so the proteinaceous part is also known as apoenzyme. It is going to be made up of protein only. And the non-proteinaceous part, or simply the non-protein part, it is known as cofactor. Now, if the cofactor is loosely bound, it is called as coenzyme. It is known as coenzyme. But if it is tightly bound, it is known as prosthetic group. Okay, so we can have a quick revision about the classification of enzymes. So they you have simple enzymes, that is the enzyme made up of protein only. It is going to have the proteinaceous part only. Example pepsin, trypsin, urease. And then you have the conjugate enzyme, holo enzymes. So they are going to have a proteinaceous part and a non-proteinaceous part. Proteinaceous part is known as apoenzyme. Non-proteinaceous part is known as a cofactor. The cofactor, it can either be loosely bound. If loosely bound, the cofactor will be called as a coenzyme, or if it is tightly bound, it will be called as a prosthetic group. Okay, so now let's continue to the next topic, that is the mode of enzyme action. So how the enzymes work. Okay, so I would like to teach you about this particular graph. On the y-axis we have the potential energy, and on the x-axis we have the progress of the reaction. Okay, so first of all, without enzyme, if you can look at here, without enzyme, the activation energy, right, the activation energy, like it is the energy required for the formation of the intermediate, okay, that is very high, correct. But when the enzymes are supplied to the system, okay, so the enzymes, they minimize the activation energy so that a larger fraction of the molecules or of the reactant can undergo the reaction. Okay, so this is how the enzymes work. So now there are two viewpoints that basically tells us how the enzymes, they are supposed to work. So first of all, the first model is the lock and key hypothesis. So just like the enzymes are highly specific, they are very specific. Okay, so the lock and key hypothesis, it basically mentions like the way a particular lock has a particular key, okay, and the key completely fits the lock. The same way the enzyme has its active site, and the active site is going to have a proper geometry. The geometry of the substrate will be in such a manner that the substrate with its particular enzyme, with its specific enzyme, they both will fit properly, just the way a key fits its respective lock. So this is the lock and key hypothesis. What it says, that the key substrate fits properly, like the enzyme, which is just like a lock. Okay, so basically hypothesis mentions that the enzymes are highly specific for this structure because of this, the geometry of the active site and the structure or the geometry of the substrate. Now what happens, so we have the enzyme, when it reacts, it binds with the substrate, when it binds with the substrate, we get the enzyme substrate complex. Now this enzyme substrate complex, the active site, the functional groups over the active site will break some bonds, or will help to create some new bonds, right, and this will get converted into enzyme product complex, and this enzyme product complex will disintegrate into enzyme plus product. So this is going to be the reaction for the lock and key hypothesis. Now we have another viewpoint about the activity of the enzyme, that is the induced fit theory. Now the induced fit theory says that the active site has two components. First we have a buttressing site, and then we have the catalytic group. We have the buttressing site and catalytic group. So now the point is, like, see, the number of the different substrates that we have, it is higher than the number of enzymes that are there in the biological system, right? So the number of different types of substrate, it is higher than the number of enzymes, the different enzymes there are. So this basically shows that the substrates which are similar in structures, they can bind to the same enzyme, right? If there are two structures and if they share some similarity in the structure, so they have the possibility that they can bind to the same enzyme. So this basically emphasizes, like, this particular theory provides us with an idea that there can be some flexibility when it comes to the specificity of the enzymes. Okay, so buttressing site and catalytic group. So let's say I'll draw this. Let's say this is a functional group, or this is the active site. So active site means here I have the catalytic group. Okay, so this is the catalytic group and this is the buttressing site. Okay, so this is the buttressing site. Let's say I have a substrate like this. Okay, so this is my substrate. So now when this substrate, when it will go and it will bind to the buttressing site, what will happen? There will be some conformational changes in the active site of the function of the enzyme, and the catalytic group, it will come closer to the substrate. Okay, so let's say this will become something like this. So now as the substrate binded to the active site, it brought some conformational changes in the structure of the enzyme, and the catalytic group was brought close to the substrate. Okay, so if you can see it over here, okay, now the catalytic group will actually catalyze the reaction that was supposed to happen. Okay, so here I have a proper diagram for this. So if you can see, the shape of the active site is different from that of the substrate. Now when the substrate, when it approaches the enzyme, when it binds to the buttressing site, there are some conformational changes in the enzyme so that the substrate can fit properly. Okay, the catalytic group will perform all the desired reactions and finally the product will get separated. So this is what the induced fit theory mentions about. Okay, so that's all from my side. I hope everything is clear till here. Now in the next class, I'll tell you about the classification of the enzymes, and also we'll talk about the properties of the enzymes, the factors that influence the enzyme activity, and finally, if we'll get some time, we'll also practice some questions. Okay, so that's all from my side for today's session. Good night, everyone. Take care. If you have any doubts, you can drop them in the comment section. Good night. Take care.